Hello, my name is Beatriz Llamusil. I am the CEO of ARTHEX Biotech, a spin-off company of the University of Valencia that is dedicated to the development of anti-miR oligonucleotides to treat myotonic dystrophy patients. Let me introduce you my team. We are a small team and we have a dedicated lab in the University of Valencia, some people helping me at the management level. And in the board of directors, I should mention two VCs on board, In Vivo Capital and ARTHEX France. And as part of our scientific advisory board, we have international experts in the synthesis of oligonucleotides and the design of antisense molecules, such as Ramon Erija and Eric Markusson, and two clinicians which are very well known in the field of myotonic dystrophy, Nicolas Johnson and Guillaume Basset. The company was created at the end of 2019, but I was personally involved in the research against myotonic dystrophy since 2007, when I initiated my first postdoctoral stay in the University of Valencia. About the disease, you probably all know more than myself. Just to let you know that although it is considered a rare genetic multisystemic disease, it is still highly prevalent. It is in fact the most common muscle atrophy in the adults. And there is no dedicated treatment, although the genetic cause is very well known. It is a very complex disease from the genetic point of view, but just to make it short, it is generated by a mutation in the DMPK gene, in the case of DM1, which is causing a toxic RNA, because this extra long RNA is folding into a hairpin here in purple that is retaining MBNL proteins. And these MBNL proteins are important regulators of splicing, that when they are retained here, there is a lack of function of these proteins that is originating part of the symptoms. And it has been reported in several models of the disease that the expression of this protein could be therapeutic. So our approach was to generate a first-in-class RNA therapy for the treatment of this disease based on upregulating the levels of MBNL proteins. As you know, most of the companies are focused on this part of the problem. They are trying to decrease or inhibit the interaction between the protein and the RNA, or directly destroy this toxic RNA. But studying MBNL proteins, we have discovered that the levels of this protein are downregulated by two different mechanisms. They are attached to the repeats, but they are also blocked by microRNAs that are inhibiting the production of this protein. These microRNAs, MIR23B and MIR218, we observed them upregulated in muscle biopsies and also in different models of the disease, and they are blocking the production of MBNL. So what we have discovered is that just blocking these microRNAs, we could increase the general levels of the protein enough to see a relief of the symptoms. So although we will still have some protein attached to the foci or to the toxic RNA, there will be a general upregulation of the protein, so there is more protein available to perform its function. To block these microRNAs, we used anti-miRs that are single-stranded RNA, and we obtained proof of concept of this therapeutic approach in two papers already published and one that is about to be published, where subcutaneous injections of these molecules produce a long-acting therapeutic effect in a well-known mouse model of the disease. So although the results with these anti-miRs were promising, during the last year, we have put a lot of effort in improving the efficacy of these anti-miRs and finding the best molecules to move into the clinics. This is the screening we performed. We started with more than 100 different anti-miRs, which were naked molecules or molecules with different hydrophobic, conjugated to different hydrophobic moieties, to improve their effects into the muscle tissues. We started with the screening in vitro, and then we moved to in vivo using the HSALR mice. So we first tested the molecules at a very high concentration to get rid of those that can be toxic. And then we tested them at a reduced concentration at just 3 mg/kg to only observe an effect in those that are highly efficacious. With these results, we selected two candidates, one molecule against MIR218, which is our main lead, and another backup against 218, that we will move forward just in case, that we observed any toxicity with the previous, with the first molecule, with the lead against 23. Just to show you the comparison of the new molecules with the previous ones, these are the molecules that we have previously published, these antimers conjugated to cholesterol. With these molecules injected at 40 mg/kg, we obtained a normalization of the strength in the mice model and a decrease in the myotonia of up to 50% maximum. However, with the new molecules, I show you here some the effect of some of the new molecules in the myotonia and in the percentage of force of the mice. As you can see with the lead candidates that I have encircled here in red, the one against 23B, and in green, the one against 218, with these two leads just injected at 3 mg/kg, we obtain even better results than with the previous molecules injected at 40. Here you can see a decrease in the myotonia of up to 75% and also a normalization in the strength. So the new molecules are at least 10-fold more efficacious than the previous ones. Testing the effects of antimers in human cells, we performed RNA-seq studies. You can observe here a group of splicings that are altered when we compared control and DM1 cells, myoblasts. When we treated DM1 myoblasts with antimer 23B, as you can see, there is no longer points in some of these effects, which means that there is no longer difference between the control condition and the DM1 treated. That means that we are normalizing all these splicings and improving the effects in some of them. When we compare these results we obtain with the cells with the data previously published by Wang regarding the alterations in the different biopsy from muscles of patients, what we observed is that our antimers could rescue about 85% of these splicings that were commonly altered between DM1 cells and biopsies in humans. We have also some data with this, with the lead against 23B in rats. We injected this compound once weekly during 3 weeks in rats at 2 different concentrations, 8 mg/kg and 30 mg/kg. And we have observed that there is, looking at the gastrocnemius muscles of these rats, we observed that there is a strong upregulation of MBNL protein. After these 3 weeks of treatment, we really have 5-fold of protein. Already with the lowest dose, we obtained a very strong upregulation of the protein. We have also tested the difference between intravenous and subcutaneous pathways or routes of administration. And as you can see with both of them, here only after 1 injection, there was no difference. So subcutaneous could be also an option for this treatment. And looking at the effects in time after just 1 single injection, 1 day after the injection, we had levels of the protein, of MBNL1 protein, of more than 2-fold. There was a peak after 5 days, but still after 22 days, we still see around 3-fold of protein in the muscles of the rats, which means that our molecule has a strong effect in the muscles. We quantified the levels of the molecule at these time points in the rats, and we observed that we were in the low nanomolar range. We even needed to test different bioanalytical methods because we were not able to detect them up below 50 nanogram milliliters. So that means that we only need small amounts of this compound in the muscles to have a strong effect on MBNL proteins, which has been directly linked to the symptoms of the patients. So well, we are moving this compound forward into the clinics, And this is our business model. We are right now in the preclinical non-GLP phase, just initiating the preclinical regulatory phase that we will move forward during the next year. Probably we will be entering into the first in human by the end of 2022, and our idea is to perform a phase 1/2A during 23 and 24, and then go to phase 2/3. That could be registrational, maybe finished during 2027. Ideally, we will be initiating treatments against other indications also during this next year, and we will also focus in the field of rare neuromuscular diseases. Thank you very much for your attention, and enjoy the rest of the talks.